health also. And therefore, in last few years, you must have seen that there are now a lot of talks going on green technology and sustainable development and green environment, and those kind of things are going on. And in this regard, I suppose this kind of courses and will be very helpful for making it sort of awareness in all the society that whatever the development we are doing, it has to be sustainable and it has to be environmental friendly also. I'm not a man from chemical engineering, so I don't know much about the synthesis and chemical processes and getting the products using these nanoparticles and other things. I'm from mechanical side, so I can tell little bit about mechanical that whenever there is a sort of a we talk about the development in maybe in IC engines or in, even in electric vehicles or any machining processes, then we want to be very careful about the carbon product generation, and then there is concept of the carbon credit. So this is what we are going on, and we try to see that the emissions which are there that are within control, and so there are various agencies are also there, then various norms are there, and new norms are coming, so as to see that there is a reduction in the emissions, particularly the carbon emissions and the corresponding products that has to be minimized. So these are the things that we are trying, and there I know that some of the nanoparticles are being used in the fuel so as to minimize the pollution. So these are some of the things that at the mechanical side we are doing. Then, as far as our institute is concerned, we are working on nanoparticles. There are a lot of departments like chemical engineering department is working on nanoparticles. Then we have got the metallurgy and material science department, who is very good, and they have got a good research team who is working in nanoparticles and their applications. Then electronics department is also there. So all the physics department. So they are all working together, and they're trying to find out the use of the nanomaterials and the products developed in that. So at the institute level, we are also very, you can say, alert about the green development and the green energy. I just like to share with you that we are having about 214 acres of green campus. So it is not just green in terms of the greenery and trees, but it is in true sense it is green. Engineering point of view also it is green. We are having about on 17th of our buildings. We are erected a solar panels, rooftop panels, and I am very happy to share with you that we are generating 1 megawatt electricity by using the solar panels, and because that is the maximum that we can generate as per the rules and regulations of the Maharashtra and I think the central government. So maximum we are developing, and I am happy that about 70% of our need of electricity we are satisfying by using the solar energy, which is a clean energy. And I am also uh, I want to share with you that this year, in last 12 months, we have saved about 1 crores 4 lakhs rupees on electricity by using the solar panel. So this is sort of an achievement that we are looking forward. Similarly, we are also developing STP, sewage treatment plant, for the recirculation of our water, and the STP of 600 kiloliter per day that has been established. Almost it is on the last phase. Maybe because of this COVID-19, it is little bit delayed. But I am sure that within 1 month that STP will be ready, so that we shall be also utilizing our water very optimally. Then we are also going for this waste food treatment plants, that is also on in progress, and within few months that will also be ready. So we are also trying from our side not just to talk but also to implement these kind of things so that our campus become green, and we can utilize the various technologies which are developed or which are being developed. So I'm very happy that this FDA is on a topic which is very relevant in today's world, and I'm happy that there are so many faculty members who are attending this, and there are the experts from various universities, various CSR lab, IITs, and Professor Ashok Kumar is also there, who will be talking to you people. And so I am sure that there will be a lot of good deliberations during this program, and it will be definitely helpful to the all new faculty members who have just joined or who are young. So it will be a sort of a enriching experience for them to enrich their knowledge, particularly in the area of the nanomaterials and the synthesis of nanomaterials. So once again, I welcome you, and I'm looking forward for Professor Ashok Kumar to come to VNIT in due course of time for signing the MOU with the Melbourne University. I virtually welcome you, sir, and then
Thanks,
when
sir.
you shall be coming over here, the
Thank
actual
you.
welcome will be there. So once again, I welcome all of you, and thank you, Professor Sonawane, for organizing this event. Thank you very much.
Thank you.
Sir, very thankful to you also because your moral support will enhance our morals, and always you boost us, to do something in the field of research. I am also thankful to Ashok sir, since particularly Ashok sir is Vice Chancellor of Melbourne University. So
Yeah, yeah.
it's a good opportunity for us, and it's a very popular university in the world. So I will take this privilege to have a MOU with Ashok Sir's University. So when JN course will be there, Professor Ashok Sir will be visiting here. So that time we will have that particular element. So and accordingly, then we will visit there in Australia in Sir's University, so that we can hand over the MOU to them, and we can do a lot more research. Thank you very much, and thank you very much, AICT peoples also. So, as a chairman, Professor, I will request you to open this FDP for participants and for all the sessions.
Am I supposed to? I have to do it?
Yeah, because you are chairman of this FDP,
Okay, okay, okay.
patron for us.
Yeah, thank you, Sonawane. I officially declare this FDA Faculty Development Program open, and I wish all the best to all the participants and the organizers. Thank you very much.
Thank you very much, sir. Professor Ashok Kumar, sir. Dear participants, now we will have a very fruitful and very important talk as a part of start of this particular faculty development program from our respected Professor Ashok Kumar, sir, who is the Vice-Chancellor of Melbourne University. I will just read out Professor Ashok Kumar's first name in a brief. Professor Muthu Pandey Ashok Kumar is a physical chemist specialized in sonochemistry. Teaches undergraduate and postgraduate students, and he has a 20 years experience in the field of research, development, and teaching. Uh, as far as Sir's age index and research profile is concerned, it is it is too much. Sir, you are awesome. I will only say, sir, you are awesome. And Sir has many several awards on his names, several fellowships on his names, including the Green Gold Prize in Industrial Chemistry. He is a fellow of Royal Australian Technical Institute since 2007. So I will request Professor Ashok Kumar sir to start his keynote and first session of this faculty development program. Professor Ashok Kumar sir.
Thank you. Can you see the slides?
Yes, sir.
Yeah.
We can see the slide.
Okay, thank you. Respected director, thank you for your invitation and also for hosting this event. And I'm sure that in post-COVID, we will be able to travel, and then I can visit you and give the Gian lecture course.
Thank you, sir.
Then we will take it forward in terms of the collaboration between our institutions. Um, Everyone, thank you, and it's good morning to you, and good afternoon to me in Australia. So today, first of all, I would like to thank Professor Sonawane for giving me this opportunity to talk to you about my research. And my research is primarily involved with ultrasound for various applications. But the topic that I have chosen today is to discuss about the green technology part of the ultrasonics in food and bioprocessing applications. Hopefully you can hear me well. If there is any problem, please let me know. Before I talk about my research, I would like to introduce the University of Melbourne to you. As I was introduced, I'm a professor in chemistry. So I do normal teaching, primarily physical chemistry. But at the university level, I'm also the Assistant Deputy Vice-Chancellor International, where I look after the international relationship between our institution and the global institutions. And recently, I've been focusing in India in terms of collaboration. About the University of Melbourne is about 160 years old, second oldest university. It was established in 1853. And we have the university has generated a large number of leaders in various disciplines. So some of the early graduates you can see appearing here. The Melbourne University has 7 campuses, but the main campus is the Parkway campus, which is in the heart of the city. I'll show you a picture of that. The rest of that's where most of the teaching happens. The rest of the campuses that you see here are more purpose-built. For example, we have in Werribee the veterinary science campus because you need a bigger land. And for example, in Burnley, mostly the agricultural part of kind of thing. So each of these campuses will have different disciplines involved, but the Parkville campus is the main campus, which is in the city. And we do have most of the 99% teaching happening in this campus. So the photograph that you see here is the campus from the city. So this is the city center that you see. Back there is the Bay Area or the Sea Area. So the Melbourne University campus, which is this park, is located in the north part of the city. And most of the departments are here and also a little bit over here, biomedical precinct. And these places that you see here are the colleges. The colleges here are equal to our hostels. So most of the international students are interstate students will be staying here, walking across the grounds to reach the departments for lectures and other things. Some of you may know the Melbourne University is the #1 ranked university within Melbourne, within Australia. Also number 32 globally based on the Times Higher Education ranking. We have about 70,000 students on campus. About 40% of them are international students. So you can see that we have a large number of international students studying at Melbourne. And the Melbourne University investment in terms of research is about 1.2 billion Australian dollars every year. Has a large community, alumni community throughout the world. So this is the entrance to the campus, the main campus, Parkville campus, where as I said, most of the teaching happens. And some of you may have noticed that we know that Australia belongs to the Aboriginal community. So we want to kind of acknowledge that the university is built on the land occupied by the Aboriginal people. So this term here, Romanjika, is welcome in Aboriginal language. As I already mentioned to you, we have large cohort of international students studying here. Coming to my group itself, I have a large number of introduction only. Within my group, we have a number of postdocs. The red ones are completed ones. The current.
ನೀವು ಒಳ್ಳೇದಾಗಲ್ಲ ನಾನು ಹೇಳ್ತೀನಿ ಅದು ಬಾಳೆಹಣ್ಣು.
In addition to that, we have both students and master students completed.
ಅದು ಎಲ್ಲೈತಿ ಇವಾಗ ಹೇಳು, ಅದು ಗಣಪತಿ ಪ್ರಸಾದ ಹಾಕಿ ನಾನು ಹೇಳ್ತಿದ್ದೀನಿ, ನಿಜ ಹೇಳ್ತಿದ್ದೀನಿ ನಾನು ಇವಾಗ ಎಲ್ಲಿತ್ತು.
Now I'm trying to go into my research up. We'll be talking set about the applications of ultrasound. Ultrasound is as a green technology mainly because it does not use the solvents like toxic solvents in many applications. In terms of the use of technology for food and bioprocessing, it is developing. Some industries are taking into their plants. So this is one of the developing areas of technologies that, in my opinion, the next 10 years or so will be widely used in industries, primarily the food industry, but also in bioprocessing industry. Instead, the green technology mainly comes from the fact that it uses less energy, more solvent or less solvent, toxic solvents. So it is an accepted technology. It's a green technology. Before I go to the applications part of it, I would like to just kind of maybe spend about 20 minutes or so on the fundamental aspects of ultrasound. And without understanding the fundamental aspects, it will be hard for you to appreciate the applications of ultrasound. So in terms of material synthesis or food processing, developing nutritional food, and so on, if I don't show you the basic part of the ultrasound, then you won't be able to appreciate. So I'm just trying to first go through the basics of the sonar chemistry or ultrasonics part. And in terms of this figure is just kind of pictorial thing to show the frequencies of ultrasound. And most of us know that when I talk, the frequency that if I can generate is in between 20 hertz up to 20 kilohertz, then you can hear it. So basically, the humans can hear in the frequency range 20 hertz up to 20,000 hertz or 20 kilohertz. Below 20 hertz infrasound, not many people can hear, but animals do use for communication. Above 20,000, which is called ultrasound, that's where most of the results that we are going to look at, or the applications that we are going to look at, uses the ultrasound, which is beyond the human hearing frequency range, about 20 kilohertz. Okay, but the ultrasound is also used for communication by animals. And most of us know ultrasound through imaging our body. For example, if someone has a baby, then they use ultrasonic scanning to look at the growth of the baby. Or if you have a tumor or any other problem in the body, people tend to use ultrasound imaging. Okay. So as kids, we used to play with bubbles. As adults also, we used to play with bubbles. But what happens when you have bubbles and ultrasound interacting, then a process called cavitation happens, which is what we are going to look into the details. So basically what we are looking here is that ultrasonic waves, when they interact with bubbles in a liquid, then a process known as acoustic cavitation happens. And that's what I will be discussing in the next 10-15 minutes. So basically what happens is the bubbles that are present in the solution are forced to grow by the acoustic field or ultrasonic field, and then they're forced to collapse. This collapse is the one that generates a large amount of energy, which can be used for chemical reactions. As I mentioned, a sound wave or an ultrasonic wave is simply an alternating positive and negative pressure wave. So when you have a small air bubble that is present in the liquid, then when the liquid around that bubble experiences the growth part of the sound wave or the expansion part of the sound wave, then the pressure around the bubble decreases. That means the bubble is growing. When the compression cycle comes through, the bubble shrinks. And when the next wave or next kind of expansion wave comes through, the bubble expands and collapses and so on. So basically what you can see is when the pressure wave is changing from positive or compression to rarefaction negative, then the bubble is oscillating. But not only that the bubble is oscillating, growing and shrinking, but also it is growing in size overall. So compared to where you start, the size of the bubble is a little bit larger. And that process, which is called rectified diffusion growth process, is specific for the ultrasonic field, where the chemistry starts to happen. So what it means is, if I want to create a bubble, thermodynamically, I can look into how much energy that I need. So for example, if I want to create a bubble in a liquid, this equation can be used to calculate the amount of energy that I need to provide to a solution. So the negative threshold, pressure threshold, which is called Blake threshold, is related to the surface tension of the liquid and the size of the cavity that you try to form. Okay. In simple terms, don't worry about any of these values. If I substitute the surface tension value, for example, in water as 72 millinewtons per meter, and then if I want to create a cavity which is of van der Waals distance, few angstroms or less than a nanometer, then the amount of energy that I have to provide is quite large, about 10 to the 9 watts per square centimeter. But in real term, you do not have to provide that much of high energy to generate cavitation bubbles, because bubbles are already existing in any liquid. So if you take water, you just shake it, you will see bubbles being formed. So what it means is you do not have to create a bubble. Simply, you need to grow the bubble that is already existing by means of this process called rectified diffusion. Okay, that is pictorially shown here. So what happens is when you have the negative pressure wave coming around the bubble, the size of the bubble increases. That means the surface area of the bubble increases. And during this expansion process, the pressure inside the bubble is very low. That means gas molecules and the solvent molecules will try to evaporate into the bubble or diffuse into the bubble. Because you have much larger surface area, more gas molecules and more solvent molecules will get inside. When the bubble is shrinking, the pressure inside is high, molecules will tend to come out, but because the surface area here is small, the number of molecules that is getting out will be very, very small. Because there is more molecules getting inside, less molecules getting outside, that's why the bubble seems to be growing in size through this rectified diffusion process. So what happens is, I'm not going to go through too much into the details. For those who are interested, there are some theoretical papers that are available that you can read. There is also another thing called shell effect, which is a bit complicated. I'm not going to go through that. Even with the area effect, you are able to understand that the bubble is growing in size over a period of time when the bubble is oscillating. What I can show is whatever we are predicting here theoretically or speculating here can be visually seen by means of visual observation. So what you see here is a capillary. That capillary has certain diameter, in this case about 400 micron or so. There's a transducer here that will drive this bubble, so that a transducer is used to create the ultrasonic waves into the liquid. And I'm not sure whether this video clip will run. The video clip runs, then you will see that the bubble is oscillating and then growing slowly. Okay, that's as I said, the growth is due to the rectified diffusion process. It looks like the videos are, okay, it is working now. So you can see that the bubble is oscillating, and it is asymmetrically oscillating, not like a symmetric, because it is sitting on the surface of this capillary, okay? But compared to where you start, you will see that the bubble is a little bit larger now. It will be much clearer if you see this one here. You can see that now it is growing, and then it will grow much bigger very quickly. So this is a special technique that we are using to monitor the growth of the bubble called stroboscopic technique. Again, I'm not going to go through the instrumental details, but simply what I'm trying to prove here is that the bubble during the oscillation, due to the acoustic pressure wave variation, it is oscillating but also growing by means of this process called rectified diffusion, okay? What happens is when the bubble reaches a certain size, then the bubble collapses very fast. And I'm talking to mostly chemists and engineers today, and you know that if the collapse is an adiabatic process, that means no energy lost to the surroundings. That means most of the energy that you are generating during this bubble collapse can be converted into heat. That heat can be used, for example, to initiate chemical reactions. So what we are trying to calculate here is using, again, a simple thermodynamic equation, how fast this bubble can be collapsed, or how fast the bubble will collapse, so that we can see whether that process can be an adiabatic process or not. So this tau here is the collapse time, r is the radius of the bubble that we are talking about, density of the liquid, pressure of the liquid, etc. They're all involved in this equation. By putting certain parameters, for example, I'm using density of water, some pressure inside the bubble, some liquid pressure, etc. By putting some values, and I'm putting the radius as about 10 to the 5 meter, about 10 micrometer. By doing this, you can see that the collapse time is of the order of 1 microsecond. So what it means is the bubble is oscillating. When the bubble is collapsing, it collapses very fast within a microsecond time scale. And that 1 microsecond may not be enough to completely transfer the energy that you generate within the bubble. The energy that we are talking about here is due to the volume change. Again, if you look into the simple thermodynamics, W is equal to P delta V. The delta V term is the one that we are talking about here. When there is a volume change, there is a work associated with that. But if the bubble is not doing any work, this work is converted into heat, okay? So in this case, because of the collapse being adiabatic or near adiabatic process, the energy is preserved, and that is the energy that is generated to do chemical reactions in our case, okay? Example, again, by using a simple thermodynamic equation that's shown here, I can calculate what kind of temperature can be generated when this bubble is collapsing in water. Again, there are some parameters here: liquid pressure, internal pressure. Gamma is the heat capacity ratio of the contents of the bubble. So if I have, for example, air, I can look at the CP, the heat capacity at constant pressure over volume, in a textbook for oxygen and nitrogen. So I will be able to calculate the gamma value. And using all these parameters, if I use, for example, liquid pressure as 1 atmosphere, etc., the temperature, theoretical temperature that I can reach when the bubble is collapsing is up to 40,000 Kelvin. But that's really very, very high. In real term, even though theoretical value is about 40,000, in real term, maybe you'll be reaching anywhere between 2,000 to 10,000 Kelvin, depending upon the experimental conditions that you use. But even if you reach 2,000 degrees Celsius or Kelvin, still it is a high temperature that can be used for initiating chemical reactions. On the other hand, because the volume is decreasing, the pressure will also be increasing. Again, using another equation, you can calculate the maximum pressure theoretically generated within the bubble. That's of the order of, say, 1,000 atmospheres, very, very high, 1,000 to more than 10,000 atmospheres, okay? So, but in simple term, when the bubble is collapsing asymmetrically, what you have is very, very high temperatures and pressures generated. It is this temperature and pressure that we generate that can be used for processing applications, including food processing and bioprocessing. When the bubbles are oscillating, they are also generating large amount of shear forces. For example, I'm going to, if this video clip runs, you will see that this bubble is oscillating, sticking to a wall of a capillary. So this capillary is about 400 microns. There is a slurry that has silica particles and some water. And what you will see is when the bubble is oscillating, you will see that the particles around the bubble are pushed very hard. So you can see that the bubble is oscillating very fast and these solid particles are moving around the bubble, but also the liquid is pushed towards one direction. Okay? Now assume that instead of these particles, you have molecules. And the molecules then will be moving very fast. And then these kind of molecular collisions may lead into faster chemical reactions. According to chemical kinetics, if you have an increase in the collision frequency, then you will increase the rate of chemical reactions. But also these shear forces will be used for changing the structure of proteins, for example, that can lead into creating some novel food materials. We'll look into all those applications later on. Also, there are other things that are happening when the bubbles are collapsing. When the bubble is collapsing asymmetrically, okay, so you can also have a liquid jet that is formed that hits the surface of this of the container. For example, the bubble is sitting on the wall, and the bubble is collapsing asymmetrically. A liquid jet, this jet has very high speed, about 200 meters per second. When that hits, it can make small hole in metal particles. Okay, that can be used, for example, to increase the porosity of metal particles for increasing catalytic activities. When the bubble is collapsing symmetrically, so in this case, we are looking at a single bubble collapsing symmetrically. The process is, the event is recorded with 100 million frames per second camera. You can see that, and the whole thing is happening within 2 microseconds. So you can see the bubble is collapsing, collapsing, collapsing. At the end of the collapse, there's a pressure wave coming out. As I already told you, when the volume is decreasing, there's a large amount of pressure generated. That pressure is now released back into the medium as a shock wave. That shock wave can also be used for pushing materials and making some mass transfer effects that can be used for applications. Okay. Just to give you an example how strong these shear forces are, either the microjet or shock waves or the streaming that we generate, I'm just going to show you one example. So this is a Navy ship that was built by the US Navy, in the 40s or 50s. They built this ship with a very strong propeller that they want to have the ship moving very fast or traveling very fast. So the propeller worked for a few days and then it stopped working. And then when they looked at the surface of this propeller, they saw large amount of damage to the propeller blades. Initially, they thought this kind of damage is due to the corrosion because seawater has salt. Salt may corrode the metal. That's what initially they thought, until a mathematician came into the picture, and then he could theoretically predict that this damage cannot be due to the salt or the corrosion. This is due to cavitation process. Here, the cavitation is due to hydrodynamic cavitation. When the propeller is moving very fast, then you can make bubbles, and those bubbles can collapse on the blades, making small cavities. Okay, and then once it was identified it is due to the cavitation, air bubbles sticking to the blade is responsible. Then what they can do is they can just simply coat the blade or the metal with some hydrophilic film or hydrophilic material, so that the hydrophobic air pockets cannot come to the come to the blade so that they can avoid the cavitation on the blades itself so that the propeller problem can be solved. So I'm again just going to demonstrate this kind of damage using an aluminum foil that I can insert into ultrasonic cleaning bath. This kind of damage is very easy to demonstrate by means of cavitation process. So this is an ultrasonic cleaning bath. I'm putting this aluminum foil into the cleaning bath, and then if I can inspect after the few seconds of sonication, I will see large amount of damage to the film. Okay, so you can see that large amounts of holes being present, and those holes are generated by the cavitation. This is acoustic cavitation. Here it is hydrodynamic cavitation, but both of them are the same kind of forces involved. Okay. Again, I can use this kind of shear forces that we generate for positive applications. So for example, in this case, I'm going to use this shear forces generated by cavitation process for making an emulsion. I have water here, I have oil here, I have an ultrasonic horn coming from the top. So when the ultrasonic horn is placed right at the interface between water and oil, then I can generate a nice emulsion very quickly. So you can see that emulsification can be done very quickly. These emulsions are very uniformly size distributed, very small size. We will talk about this later on in food emulsions, some applications. These are some equipment that are being used in a laboratory environment. For example, 20 kilohertz horn that we just saw for making an emulsion. The high frequency, like 200 kilohertz or 1 megahertz units that can be used for redox reactions, and also the ultrasonic cleaning bath that we normally use. Okay. And just for you to know, being most of you are engineers, you may want to know whether large-scale ultrasonic reactors are available. Yes. This was an early work by a group in Caltech, the way they were trying to use the cavitation process for treating wastewater. So in this case, they had 4 transducers, each of them operating around 4 kilowatt, very high power unit. And they generate cavitation in this reactor. The wastewater is being pumped, and then the contaminants are decomposed here, and then you get clean water out. So this is one equipment that is available. This is another one that is meant for the soil contaminated with oil, for example, can be treated with this kind of 20 kilohertz flow-through ultrasound unit, where each of these unit works around 1 kilowatt. So, for example, if you have contaminated soil locally, rather than you go and dig the sand and bring and then clean it and then bring it back, you can bring this kind of equipment to the local area, dig the sand which is contaminated, put it into the reservoir, mix it with some water as a slurry, pump it through the ultrasonic reactor. Then the cavitation process generates shear forces that can clean the sand, and then you just kind of filter it, and then you can refill the soil. So this is one example where ultrasound is used for treating contaminated soil. This is one of the equipment. We are going to look at the application in detail later on that we have used for processing milk. In dairy industry, there's a major problem in terms of the heat stability of milk proteins. And I will talk about this in detail later on. But here we are simply showing that we can use this kind of flow-through ultrasound unit for processing large volume of milk for various applications. Okay. And also there are other large-scale ultrasonic equipment available where the transducers are fit outside the processing tube, so that there's no any direct contact between the transducer and the liquid being processed. So in this case, it's a non-contact ultrasonic reactor where the transducers are sitting outside but creating cavitation, inside reasonably homogeneously that can be used for some, again, like, milk processing or emulsification or other applications. Okay, so now we have some sort of idea in terms of what kind of events are happening when you have bubbles interacting with ultrasound. We generate a large amount of temperatures, pressures, shear forces, and other things that can be used for certain applications. Just to give you a summary of the things that you want to remember before we look into the applications that we are going to see soon. The key aspects that you want to remember are the bubble collapse or the bubble implosion is a near adiabatic process. Large amount of temperatures and pressures are generated, and the process can also generate some radicals for chemical reactions, which I'll highlight that a little bit later, which is called sonochemistry. Sometimes light can also be emitted from these bubbles, as you can see here, because you have a hot body, it's like a black body emission, so it's called sonoluminescence. But sonoluminescence is more useful for understanding the physics of the cavitation process. We are not going to look at that here. As I mentioned, we have the shock waves, microjet, and other shear forces generated during this process. Okay. Now, the ultrasound can be used to drive chemical reactions using either the physical effects in terms of increasing the mass transfer and other things, which we will look into some examples later on. But also, we will be generating some primary radicals. These primary radicals can also be used for redox chemical reactions. But for the applications that we are going to look at, which are primarily food and bioprocessing, maybe we don't want to have too many radicals because that can also have negative impact. So I will talk about that later on. Okay. In terms of the chemistry or chemical effect, if I use water as the solvent, when the bubble is oscillating, water will evaporate into the bubble, and due to the high temperature that you generate, water will be decomposed to form hydrogen atom and hydroxyl radicals. Now, as chemists, you know that hydrogen atom is a reducing agent, hydroxyl radical is an oxidizing agent. So basically, we can do either reduction or oxidation or both redox reactions. And that's what chemistry is, okay? So in principle, we are using our cavitation bubbles as electrochemical cells to do redox reactions. Now, you may wonder, how do I know that these radicals are produced? In a normal sense, how do I quantify the production of these kind of radicals for a given system? Again, I can use some simple chemistry. The hydroxyl radicals that I'm generating in the cavitation bubbles can combine to form hydrogen peroxide. The hydrogen peroxide can be easily detected by means of iodide reaction. Iodide reacts, or hydrogen peroxide oxidizes iodide to form iodine, I2. I2 reacts with excess iodide to form triiodide complex. If I have triiodide complex, if I add a bit of starch to it, I can see a blue color. So in my system, if I want to know radicals are generated, I simply have to sonicate water in the presence of some iodide and add a bit of starch. If I see blue color, that means I'm generating radicals. That's a kind of qualitative way of checking it, but also you can, using the same technique, quantify the amount of radicals that you generate. Again, I'm trying to show you a video clip. If it runs. So here we have a small amount of potassium iodide solution, small amount of starch. So I'm just going to demonstrate the formation of the radicals within the sonication medium. So I'm pouring the potassium iodide solution into the reactor. This is a lab scale reactor. And then I start sonicating. You can see the solution is bumping. and then I'm adding a small amount of starch to it. And as you add the starch, you will see slowly the solution turning blue. So what it tells me is that I'm producing I3 minus. If I produce I3 minus, that means I'm producing hydrogen peroxide. That means I am producing hydroxyl radicals. So it's a quantitative way to check whether your system is producing radicals or not. So now with this kind of background information or fundamental details, we are now going to look into some applications, mainly in the food and bioprocessing. Now, one thing you can also notice from the previous slide that simply when you generate this kind of physical forces or chemical forces, we are not adding any chemicals to the medium. We simply take water and sonicate. Okay, that's where we are calling this as a green technology, because we don't add any external chemicals to initiate chemical reactions. So in my lab, over the past kind of 15 years or so, using the fundamental knowledge that we have developed pretty much for about 10 years or so, I started working in this field in 1996. For about 10 years, I focused on the fundamental aspects of ultrasound or acoustic cavitation. From the understanding that I had, then we could focus on our applications in various areas. For example, energy, environment, health, and food. For example, we can make materials, catalytic materials that can be used for solar energy conversion, for example, or for other catalytic reactions. Okay, so that's and that can be used for environmental applications also. We can use the processes that we are the physical and chemical processes for making emulsions and other things in food applications. We can also use them for wastewater treatment. We can use them to make biomaterials. Okay, for example, drug delivery or nutrient delivery vehicles. And also we can control some sonar processing. Today, I'm not going to go through all the applications. I'm just going to go focus on food and bioprocessing. This area here, food processing and also bioprocessing. Okay. The first one that I'm going to talk to you about is separation of fat from milk. So basically, this defatting milk or making skim milk from full cream milk. And this technology was developed by the CSIRO in Australia, in Melbourne. And the concept is demonstrated in this particular video clip. So what you will see here is that we will see how to generate a standing wave and how that standing wave can be used to separate the fat or separate water droplets. Okay, this was a TV show that was shown in Japan. So one of my collaborators, worked with a TV company to develop this clip. So I'm just going to show you and then show you the application of that. So we have a transducer here, a water droplet, a reflector here. Okay. So as soon as you start the transducer, this is like a nebulization process. You can see the water droplets are generated, and the water droplets are now suspended in air due to the formation of what we call standing wave. Okay. So as you can see here, now it is a slow motion. You are generating or nebulizing the water droplets into small droplets, and these droplets are then pulled together by means of the acoustic force, where you have pressure antinodes and nodes. So all the small droplets are now drawn together to form these big droplets. Okay, and they are suspended because now the acoustic force is holding them together. It's this application that is being used in separating fat from milk, but also it is now used in palm oil industry in Malaysia for oil separation. Okay, so what you have here is they have taken milk, they have added a bit of dye so that you can see where the fat globules are. The rose color that you see is simply an organic dye. There's a transducer here which is in megahertz range, and then when they sonicated, they could hold the fat globules. So basically, you have small fat droplets, put them together, then they become bigger. And that fusion to happen, you need a bit of time. That's what the standing wave is doing here. Okay? And as soon as you turn off the ultrasound, you can see the fat globules are separating out. And as I said, instead of having this, if you have extracted oil from plant, for example, palm oil from palm, then when you extract, you are not simply getting an oil, you are getting an emulsion, because the plant cells have water, they also have proteins, they also have oil. So you have oil, water, protein, then basically form an emulsion. To separate the oil from an emulsion is very difficult. So what the CSIRO has done is, by using this kind of the standing wave pattern, you will be able to coalesce the oil droplets by means of the standing wave, and the oil separation becomes much easier by holding the oil droplets together, as you can see here. So this is an industrial process that is being used in palm oil industry in Malaysia at this stage. We were involved in a process called the improving the heat stability of milk proteins. And most of us know that if you take milk, about 80% is water, 80 to 90% is water. And then you have 5% milk proteins, 2% to 5% some carbohydrates, plus some fat and other things. Okay? And normally, when they want to make milk powder, okay, the milk powder is prepared through a process called spray drying. So they separate the fat out, and then you have this, milk that is primarily of protein. Okay, so the protein containing milk, which is called whey protein. So the whey protein is milk protein that is pumped through a spray dryer through a membrane at very hot conditions. So first they heat the milk. The milk is heated up to maybe 90 degrees or so, and then they spray into a hot chamber where the water evaporates, and then get the milk powder. But before they are being sprayed into the hot chamber, the hot milk is coming through this tube, which is about 90 degrees or so. At that 90 degrees, what happens is sometimes you may denature the protein. Okay, so most of us know that a protein molecule has a global structure. So they have all the hydrophobic part of the protein hidden inside through this intramolecular forces. And when you heat the protein, then basically you are breaking all the intramolecular forces, and then they become entangled. I'll show you a bit more of this later on. So this is called heat-induced denaturation of protein. Once the protein is denatured, then they can easily aggregate. Once they form aggregate, then the viscosity increases, then the whole system is blocked because it becomes a gel. So this is a heat-induced gelation process that gives lots of trouble to the dairy industry. Okay, and so what they are very keen is, most of you know, if you take milk and heat it at home, you keep on heating, you'll form a thin film at the top. That is the aggregation of the denatured protein. The same thing happens here also, but in large quantity. So in this case, the dairy company approached us asking whether we can use ultrasound to increase the heat stability. What it means is, you heat the milk, they should not form a gel or an aggregate. So we did lots of work for 3 years. So what I'm showing in this diagram is the outcome of the 3 years of research that we have done in collaboration with the dairy industry. So what we are doing, we are taking a whey protein solution. It may be 5%, 10%, 20%, doesn't matter. So we are simply heating and continue to heat, okay? And then nothing happens. I'll show you the result. One process is simply heating, The other process is heating to some time and then stopping and then continue to heat. Then the third one is we do a bit of heating followed by sonication, and then we do another heating. So you can see that all of them go through preheating and post-heating. Only one of them goes through the sonication part. So the result that you see here are again the overall summary of 3 years of research. If I take the whey protein solution or the milk protein solution, if I simply heat it to 90 degrees for 1 minute, then you have a large amount of viscosity increasing. Okay, you can see the viscosity is increasing significantly. Taking that preheated one, I sonicate the solution for 5 seconds or 1 minute, the viscosity decreases. Whether I sonicate for 1 minute or 20 minutes or 40 minutes, viscosity does not change. Okay. So that doesn't show anything except that the heat-induced aggregation decreases with sonication. Okay, it does not show anything in terms of heat stability. But what is important is the next figure. So I'm taking the heated milk solution, continue to heat, the viscosity continues to increase. Okay, you can see that. Whereas if I take the 1 minute sonicated one, heated and then 1 minute sonicated, and then do further heating, the viscosity does not increase. So what we have done is by doing this 1 minute of sonication after a preheating step, 1 minute of sonication gives you heat stability. Does not matter whether you heat it after 5 minutes, 10 minutes, no increase in the viscosity. So what it means is somehow 1 minute of sonication of the preheated sample is giving heat stability to the milk protein, which is what the industry wanted. Because remember that I told you that they have to still heat the milk. So when they heat the milk, of course the viscosity will increase. And what we are simply doing is at that point, the milk is pumped into a tube in a hot condition where the viscosity is a bit high, and we are simply putting a sonication step in between. And even though it says here 1 minute, now we can do it in 5 seconds. A short sonication pulse at that point gives the heat stability so that it never forms a gel, so that the operation can be continuous. So this is a big advantage for the dairy industry when they do the spray drying process, because they don't have to worry about heat-induced gelation, because the sonication has given that stability in terms of heat stability. I'm happy to go through the mechanism, but the mechanism itself is very complex. To save time, I'm going to skip the mechanism, but if someone is interested, I can send you some papers later on. It is not just the proteins that we are giving this kind of heat stability against the viscosity increase. Even for starch, we can do the same thing. Here, we are taking certain amount of starch solution. You know that starch is heavily used in various industries, paper industries, food industries, as a gel gelation agent, etc., etc. So controlling the viscosity of starch is also important in food industry. So what you are seeing here is that when you take starch and heat, the viscosity increases. Okay. Whereas when you heat the starch after certain sonication, so here, for example, we are sonicating for different periods of times. So you can see the blue curve, the viscosity is not almost changing. Okay. So not only that we are giving heat stability to the proteins, but also we are giving heat stability to the carbohydrates also. So it's kind of general process due to the shear forces. It's mainly the shear forces that we generate are useful for this thing. So this kind of high shear forces can be generated at low frequency, So low frequency ultrasound, this is particularly 20 kilohertz, can be used to control the heat stability of proteins and starches. Okay. And now I'm going to show you an application in food emulsions, making food emulsions using the shear forces. And most of you know that if you take milk, milk is an emulsion because it has fat, globules, and water. The fat globules are stabilized by proteins. Ice cream is an emulsion, cheese is an emulsion, and so on. So I'm just going to show you how good ultrasonic emulsification process compared to a conventional ultra-Turex or homogenizer, which is used for conventional emulsification at an industrial scale. So here what we have done is we have taken skim milk and we are trying to increase the nutritional value of the skim milk, okay, by adding certain amount of fish oil, or in this case, omega-3 oil, flaxseed oil. That's what we have added here. About 10% of the omega-3 oil is added to increase the nutritional value of milk. And because oil is not going to easily mix with milk, we need to emulsify that. So we are now comparing emulsification using an ultra-Turex or homogenizer and a 20 kilohertz ultrasound. Okay, and what you can see is after you disperse the flaxseed oil in skim milk, and then you look at the stability of that emulsion, you can see that this looks like milk, whereas here you can see an yellow color, which means the emulsion is separating out. Here it is only sonicated for 5 minutes. Here it is sonicated for almost like 15 minutes. Even though we sonicate this for 15 minutes, the emulsion is not stable. Okay, and also we are making much smaller size droplets also, which is important for certain food products. So the question here is why the emulsion droplets are very stable in a surfactant. Again, it is a chemical free. We don't add any stabilizer. Okay, without adding any stabilizer, that's why we call this as a green process. Why an emulsion is stable when you make it using ultrasound compared to the ultra-Turex? That's because we are making the stabilizers from the protein itself. Okay, so now I'm trying to show you what happens to a protein, like a milk protein, for example, or egg protein, for example. Normally, the proteins will have this kind of globular structure. When you heat the protein, they become inactive because you basically entangle them due to the breaking of the intramolecular forces. They become much elongated, and once they are elongated due to the hydrophobic interaction, they start interacting with the next neighboring proteins to form a proper gel or a solid. So in this case, like when you fry an egg, that's what you do. You convert the egg proteins denatured, and then it is irreversible. Whereas if you take the protein, like in milk protein, and slightly warm it, the protein becomes temporarily denatured and not completely heating it. It's warming, okay? And then if you leave it for some time at normal temperature, the proteins go back to the original state. So this is a reversible process. So this denaturation is a temporary denaturation, not a permanent denaturation. Here it is permanent denaturation. So what the sonication does is, sonication does temporary denaturation of the dairy protein. Okay, so you are sonicating for 5 minutes. During that 5 minutes, you are temporarily denaturing the protein. When you temporarily denature the protein now becomes a stabilizer because it is surface active. So when you generate an emulsion droplet, the protein goes and sits on the top of the emulsion, make it stable. Okay, so without any external stabilizer, we are creating internal stabilizer to stabilize the emulsions. Now you can ask me this question, if you are going to partially denature or temporarily denature, if they are all going to stabilize the emulsion, then are we losing the protein? which is also nutritional for our health. What we have done is we have quantified the amount of proteins that are denatured, that are used as stabilizer. It is only 0.1% of the total protein. The rest of the 99.9% of the protein return to the globular state after some time. So the sonication is almost like warm heating. It's heating like a warm, very, very low temperature heating, where it also makes the process reversible in terms of the denaturation of the protein. The 0.1% denatured proteins act as stabilizer, making the emulsion droplets stable. Okay, so what I have demonstrated here is that the ultrasonic processing is a green process for making an emulsion to make them stable without affecting the quality of the proteins in milk. Okay, in fact, this process simply increases the nutritional value of the milk by adding an antioxidant like an omega-3 oil without affecting the quality of the milk. So that's the advantage of using an ultrasonic process for making an emulsion. The same process has been extended to form a double emulsion to reduce the fat content in cheese. You know, most of the Western countries, they like cheese, and cheese has large amount of fat. So again, in collaboration with the dairy industry or a cheese company, we were trying to make a cheese with less fat without affecting the quality of the cheese itself. So what we are doing is a process called double emulsion using ultrasonic emulsification process. We take the skim milk, so this is the skim milk part of it, and we add a large amount of oil, which is the fat in this case, and then we create water in oil emulsion. Okay, so we make a water in oil emulsion, and then we take that water in oil emulsion, disperse that on top of large quantity of milk, and then do another emulsification process to create a double emulsion. So here you can see that I have water in oil in water emulsion, double emulsion. So if it is a normal process, this yellow part, the oil droplet, will be full of fat. By doing the process that we have followed, half of that fat is converted into water. What it means is the cheese that we are going to make at the end will have half water and half fat. So you have reduced fat. Same process can also be used to reduce the fat content in chocolates, for example. Okay. And this is just some images to show that the water droplets that are encapsulated, they are very stable inside the fat globules. So you can see those water droplets. This is the cryo-TEM image. You can see water droplets are still there. So what it means is the process that we are following in the double emulsion makes the water droplet stable, and then we use this fat globules that are half filled with water to make the cheese. And end of the day, we can show that the cheese that you make out of this double emulsion method that we use looks similar to the cheese that is commercially made with full fat. And then we also did some sensory analysis and other things. So they all can, satisfy all the properties. Here, for example, we are showing the stretchability of the cheese that we are making. For example, this kind of cheese is used in making pizzas. You can see that this is made of double emulsion fat that we made in our laboratory, and it is quite stretchable. It is not affecting the quality or sensory values of the cheese that we are making. Again, some more proof that these water droplets are still stable after we make them. So there are two examples for the food, and I will take one more example and then move into biomedical applications or nutritional applications, which will take maybe another 15-20 minutes, and then I will give you enough time for asking me questions. So here what we are talking about is making some polyphenolic nanoparticles for food applications, but also can be used for drug delivery applications. Okay, so they are used in both in food applications for delivering nutritional food, but also it can be used for delivering drugs in targeted drug delivery applications. I will highlight both of them. Now, polyphenols, most of you know, are very useful for your health. They are, present in most of the fruits, vegetables, for example, and they are used in a number of applications, biomedical applications, food applications, cosmetics, and curing some diseases, and also in dye industries. So they have the polyphenolic molecules are useful in large number of applications. The best option for you to take good quality polyphenols or antioxidants is by eating fresh food and fresh vegetables, fresh fruits and vegetables. But many people around the world, they are not fortunate enough to have fresh produce. So for them, normally what we do is we convert these kind of polyphenolic compounds in fruits and vegetables into capsules, and then you can eat these capsules on a daily basis to get the nutrients that you need for your body. When you make this kind of nutritional capsules, what is important is that when you eat these capsules, they have to release these polyphenolic compounds in your stomach, and then these polyphenols have to stay there long enough so that your intestinal tissue can absorb those kind of polyphenolic compounds, and then they get into get into a system to give you the benefits that you need. And there you need to worry about large amount of chemistry in terms of absorption properties of these polyphenolic compounds and stability of these polyphenolic compounds, functionality of these polyphenolic compounds, etc., etc. That's where we are working on in recent times. One of the polyphenolic compounds is tannic acid. Tannic acid is now being researched, and a large number of research articles are available. That's because they have, as you can see, large number of polyphenolic units or flavonoid units that can be used for antioxidant purposes. And what we have done is we have started working with some simple phenolic compounds and see if you can convert these simple phenolic compounds into nanoparticles. And once you make them nanoparticles, then you can functionalize them so that they can stay in your stomach for a longer time when they are released. So it was one of the proof of concept studies that I'm trying to show you some results. So the compound itself is called BTT. This is part of your the proteins in our body. It has this tyrosine moiety. So when we sonicated this sample in water, what we noticed is they formed this kind of nice nanostructured material. Okay. And then when we did some kind of chemical analysis, what we noticed is these compounds have been converted into higher molecular weight compounds. So, for example, we found dimers, trimers, tetramers, etc., using mass spec. But also, we noticed that we generated some hydroxylated products and also some dimer and trimers either in HPLC or in mass spec. And in terms of the mechanism, again, I'm not going to go too much into the detail. Basically, the chemistry behind this process involves hydroxyl radical abstracting a proton from this benzene ring to form a radical. That radical then can form biphenyl linkage, and then that can grow into trimer, tetramer, etc. Okay, so basically what we are doing a simple polymerization reaction induced by the radicals that we generate that leads into oligomerization. The oligomers, the number of monomers in the oligomer keeps growing until it reaches the solubility limit, and then they all collapse into this kind of nanostructures. Okay. For this process to happen, it is a high energy process. So not only that you need the hydroxyl radicals, but also this reaction needs to happen at the collapsing bubble interface. When the bubble collapses, we generate large amount of temperature, and the temperature is leaked out to the interface. So the interface is a little bit hotter than the solution temperature. the higher temperature here and the hydroxyl radicals that we generate with them when the bubbles are collapsing together drive this kind of nanoparticle formation through polymerization process. Okay. Again, if I want to do this reaction chemically, I need to add solvents, I need to add some kind of initiators, etc., some external chemical species. But in ultrasonic process, it is clean process, green process. No chemicals added, simply water with this particular chemical, which it's not even a chemical, it is a compound that is present in your body. We are converting that compound that is present in your body into a nanoparticle that can be used for delivering. For example, this itself is having some nutritional value, but also I can encapsulate some drugs. So if I add some drug into the solution, the drug will be encapsulated within this nanostructure that can be then delivered into different parts of your body. Okay. And as I said, we also looked into the tannic acid itself. Tannic acid also went through this same kind of reactions. In fact, tannic acid went through a different pathway, but still led to the formation of kind of large particles like this, different sized particles. Some of them are micron-sized spherical. Some of them have disc shape. So different shaped particles could be made. They all had different optical properties. So we could use them for biomedical imaging and other applications. Okay. So I'm just rushing because I need to cover a few more. But simply what I wanted to show was that you can make some nanostructures or microstructures that have some nutritional value, they have some biomedical value for either therapeutic or imaging or diagnostic applications that can be used for in various applications. Again, simply by sonication, no addition of reactants or no addition of external chemicals. The last bit that I'm going to kind of cover is, again, looking at some microspheres or coastal structures that can be used for either nutrient delivery or for drug delivery by simple sonication process. So here we can take some proteins like milk protein or lysozyme, which is present in egg and other materials, food materials, make them into spherical structures. Within these spheres, we can encapsulate, for example, fish oil or vitamin and other things that can be used for improving the nutritional value of the food. So what we are doing is we are simply taking fish oil, for example, and then forming an emulsion of that fish oil using a protein. But in this case, we are cross-linking the proteins to form a thin shell, as you can see. Within the shell, we are putting this liquid, which is the emulsion droplet that you initially form. So what it means is that if I take fish oil and if I make an emulsion of that fish oil in a protein solution, and if I try to simply add that emulsion into a product, food product like yogurt or milk, then as soon as the emulsion becomes less stable, or say if the stabilizers are going out because you are now putting into a different matrix, then the internal core, like the fish oil or vitamin, will diffuse out. Once they are out, then they interact with oxygen, which is in the food material, and then they get oxidized, they lose their functionality. But when they are protected by a thin shell of protein, oxygen cannot diffuse out, the material cannot diffuse out until you eat, where it can be delivered. When you start kind of chewing the product, or when the product goes into your stomach, the forces that are generated within your stomach can be enough to break the shell, and then the oil can leak out, which means you are delivering the functional food where you need to deliver. Now, the process itself is a bit complex. It's take a protein solution and then sonicate. So in this case, rather than encapsulating an oil, we are encapsulating air. But instead of air, you can also encapsulate oil. But this is just to demonstrate the mechanism. You take the protein goes to the interface, and then the protein gets cross-linked, and that's how you form this kind of shell. Okay. And again, I don't want to spend more time in going through the mechanism. It's more into the applications. So, for example, within these microspheres, we can encapsulate a drug. Doxorubicin is a cancer-active drug, so we can encapsulate that into a protein shell, and then you can functionalize this protein shell in such a way that once you inject this into your blood or into your body, it only goes to the tissue where it has to be released. For example, you can functionalize the shell material so that it only sticks to cancer tissue, okay? And then you can locally break it and then release the drug where it has to be released. So it's called targeted drug delivery. But in this case, we can simply, encapsulate various materials. For example, here we are encapsulating gold particles, because gold particles do have some medicinal value, including antimicrobial value. And also for bioimaging, gold particles are used in our body. Okay, so you can use a similar process for various different applications. And again, what we are good at it is to not simply showing that these things are happening, but also to understand why these things are happening so that we can have better control over the functionality and physical properties of this material. So here, we are trying to control the size and size distribution of the microspheres that we are making by changing the ultrasonic parameters. For example, when we use 1 centimeter diameter ultrasonic horn, we have much broader size distribution and also larger bubbles or microspheres. When we use a smaller one, we get a narrow size distribution, smaller size. When we use a flow-through horn, then we are making much smaller quantities. Okay, so you can see that by playing with the ultrasonic parameter, I can control the size and size distribution. If you ask why do I need to worry about this, I need to control the size and size distribution because for each application, these microspheres have to be of the order of certain size range. If I'm going to inject them into my blood, then they have to be in the size range 1 to 5 micron. If I'm going to add them into food materials, they have to be around 20 to 50 microns. So I need to be able to control the size of these microspheres. Now, what we have identified is, again, I'm not going to go too much into the details, that the cavitation zone, so this blue region shows where the cavitation bubbles are generated, acoustic cavitation bubbles are generated. So when you use a 1 centimeter horn, you can see that the cavitation zone is much larger. When you use a 3 millimeter horn, cavitation zone is much smaller. So as you decrease the size of the cavitation zone, the size distribution decreases. Also, you get smaller particles or smaller microspheres. When you take the other one, which is the flow-through horn, the horn is like this. I'm putting my camera here. I don't see anything, okay? But if I put the camera at the bottom, I can see the cavitation happening right at the tip of that horn, okay? So what we are doing is, we are converting a 3-dimensional cavitation activity into a 2-dimensional cavitation activity. And that seems to control the size and size distribution much narrower and much smaller, okay? The other thing is we need to also look into the mechanical properties of these microspheres for various applications. For example, if I'm going to inject this into my blood, then the viscosity is different. So I don't want them to be broken when I inject until they go to the right place. But also, if I want to put them into food material, for example, if we want to put them into yogurt or chocolate, then they go from normal viscosity range to a high viscosity range. Chocolate is almost like solid. So when you put that kind of high viscous medium, then these microspheres can break. That means they can release the content. That means you may lose the stability. So here we are trying to use the atomic force microscopy and laser scanning confocal microscopy together to look at the elasticity of the microsphere we make. And then we can control the elasticity by playing with the nature of the shell material, the protein that we are playing with, or biopolymer they are using. Okay, so you can see that I can squeeze the microsphere. It's like a balloon, and then it just comes back. Okay, so I can look at how strong these kind of microspheres are for various applications. And one application that we have done in collaboration with Cadbury Chocolates is to make flavored chocolates that are of high quality. If you have taken some flavored chocolates, and most probably you would have taken a product which is made through the process conventional. In the conventional process, they take this kind of flavor, mix it with silica particles, and then spray dry it. The silica particles have porosity, and the flavor will get into the pores, and then they dry it, and then they mix it with the cocoa butter, and then they make the chocolate. So if we take one of the silica particle, only 10% is the flavor, 90% is silica, basically sand, and that's what is put into the product. And also the flavor itself is heat treated. Whereas if I use our technology, the ultrasonic encapsulation technology, then basically what we are doing, we are simply emulsifying the flavor in a protein shell. For example, here you can see the picture where we have strawberry encapsulated microspheres. Here it is mint encapsulated microspheres. So once you encapsulate them, then you have pure flavor, liquid flavor inside the shell, 99.9% pure flavor without heat treatment. And the thin shell is just a protein. So that's also nutritional, okay? So what we are doing is we are avoiding the addition of sand into chocolates, also improving the quality, and also the amount of flavor that you need to add, because now we have 99.9% flavor compared to the 10% flavor in silica. The quantity that you add into your product is also lower. So it has some economic benefit also in addition to the quality benefit. And this is now it's taken by Cadbury Chocolates for production. Using the same concept, recently we received a grant from Gates Foundation. So rather than putting flavor, I can put a vitamin, for example, or I can put iron oxide, for example, and then make this microsphere nutritional. In many parts of the world, some parts of even in India, and also in other countries, children do not get enough vitamin, or they do not get enough iron oxide. Those kind of nutritional components that are missing in the common food product. Due to that lack, then they get into the kids have some diseases. Okay, so what the Gates Foundation is very keen is, if I can make this kind of microsphere filled with some nutrients like vitamin and other things, and then if you can mix them with the food product, like if I can incorporate this into chapatis or bread or biscuits, that you can store them for a year without losing its functionality, then you can send them to any part of the world without worrying about refrigeration, without worrying about the temperature, etc., because the shell is protecting the whole nutritional compound inside. Then we can kind of avoid any kind of children who are missing this kind of nutritional food. So that's the purpose of this work that we have done in collaboration with, funded by the Gates Foundation. And here, going through in detail, we incorporated some vitamins into the microspheres, and then put them into chapatis. And then we dissolved the chapatis back into water, and then showed that the nutritional compounds that we added through this microsphere addition, they are still there without losing their functionality. And now we have asked them to fund scaling up that process. Okay, so currently we are waiting for that funding for scale up process. So initially we have completed the proof of concept study. Now we are going for scale up if we get the funding from Gates Foundation. Other area that people may be interested is food waste. For example, ultrasound can be used to extract components from food waste and also convert the components into some polymeric compounds to make some useful materials. So we, in collaboration with one of the universities in Hong Kong, we are working on converting food waste into biofuels, but also some other materials. Okay. So what I have shown is, without going through much details, I have given you several examples where ultrasound-induced shear forces can be, or the physical forces can be used for making useful products in food and biomedical applications or bioprocessing also. And they're all prepared without adding any external chemicals, simply by the processes that are happening due to the interaction between ultrasound and bubbles in a liquid. And so hopefully I have demonstrated that ultrasound technology is a relatively easier technology, greener technology, less energy consumption technology that can be taken into some industries like food industries and biomedical industries to make some useful products. Okay, so at this stage, I think I have come to the end of my talk. So I'm open to any questions from you. Thank you all for listening to my talk. All of you may have some questions, but before you ask me a question, think about this question. The question that I have for you is that we are using ultrasound for imaging our body. And what we have seen in my lecture is that ultrasound can do damage to proteins, ultrasound can do chemical reactions, ultrasound do lots of different things in your body. So the question here is, how safe to scan your body using ultrasound? Think about it. If anyone has the answer, please let me know during the discussion. Otherwise, I can provide the answer later on. Thank you, and I'll wait for your questions.
Sir, very thankful for your presentation. It was very fruitful. So, few questions have been posted. One question is posted by Kumar M is by Naga Kumari. Sir, can we increase the expiry date of food by ultrasound?
Sorry?
Sir, if you see the chart, there is a questions. Can we
Yeah.
increase the expiry date of foods by ultrasound?
No, people are trying to do that. For example, they're taking fruits or vegetables and then sonicating fruits and vegetables in water to increase the shelf life. There, the idea is, when you take a piece of fruit or vegetable, you have some bacteria growing on the top. So sonication process can clean this. Okay, you can kill the bacteria, which means you can increase the shelf life. People are working on this in particular in China. But in my opinion, I think it is a high energy process, because you have to put large amount of acoustic energy for cleaning the surface of these kind of food materials. But it is doable. In terms of energy efficiency and economics, I'm not sure about it.
Sir, there is another question from one person. Can this method be helpful in destabilizing emulsion between oil and water, which may help in their separation?
Can you please read that again? Sorry.
Can this method of ultrasound be useful in destabilizing emulsion between oil and water, which may help in their separation?
So I'm not sure about the question. This question is about making an emulsion or making the emulsion stable.
Separating emulsion.
Okay. And I think that's what I talked about the oil industry example. Using the standing wave, people have tried to destabilize the emulsion So when you, for example, extract oil from a plant, so what I have shown is that by bringing the oil droplets together, by having a standing wave, you can easily separate the emulsion. And that is being currently used in palm oil industry. Sorry, I'm not hearing anything. I think you are on mute. I'm not sure. I'm not hearing you.
Sir, one question is technically, is the whole process is cost effective?
Yeah, it depends on what application we are talking about. So if I'm talking about heat stability in milk, yes, it is cost effective because I only need to sonicate the milk for 5 seconds. Okay, and that gives the heat stability. What it means is the plant can run continuously without any break. So if a dairy plant has to shut down to clean the gel that is produced, it may take a full day. So you are wasting a full day of operation by spending 5 seconds of sonication, which is a small amount of energy that you are putting. The process become efficient economically. On the other hand, if you try to use this for wastewater treatment, where you have large quantity of water, a small amount of pollutant, it may not be efficient. So what I'm trying to say is for some applications, most of the applications that I talked to today, they're all efficient, energy efficient. But some applications like cleaning the fruits, surface of the fruits, those kind of things, they may be high energy processes. So it depends on which application you are talking about. Again, I think you are muted.
Sir, during preparation of capsules with dried vegetable powder, how to decide the shelf life of or expiry of the same?
When you try to make this kind of encapsulation things, that each of them you have to do the shelf life measurements. For example, when we looked at the encapsulating vitamin or encapsulating drugs using these protein shells, we basically looked at for a year. Even after 1 year, the products were very stable. So for example, if you make a biscuit, some of the biscuits you can keep them for 2 years, 3 years, yeah.
Yeah.
And once you incorporate the microspheres into those biscuits, then they are quite stable in there because of the protein shell. So, the oxidation of the functional compounds is the major issue. And as far as you avoid oxygen getting in contact with your material, it's quite safe. So the shelf life depends on the type of the product that you deal with. For example, some of the emulsions that we make, even the emulsions alone, they are stable more than 6 months.
Okay. Sir, my question, my personal question, you have elaborated a very good example of reducing fat from cheese. So, how much percentage of fat we can reduce from cheese using ultrasound?
You can reduce up to 60% based on our study.
Okay.
But when it goes to a product, for example, we checked the sensory value of the cheese with the 60% replacement, 10%, 20%, 30%, and so on. The sensory quality is lost after 30% for cheese. But if you want to go for chocolate, even up to 60% it may last.
Okay. Sir, there are studies, and I have also studied extraction of tannic acid from tea, and you have also elaborated good example. Because generally, as far as herbal tea is concerned, This particular example is very good. Extraction of tannic acid from tea. Because of tannic acid only, we felt acidity and many types of side effects in the human body. So we have extracted tannic acid using different solvents by performing soft slick type of apparatus. Very simple, but you have explained in a very good way by ultrasound. We have also tried ultrasound. At what percent we can we totally remove tannic acid from tea?
I have not tried this. I think there are two different things. Tannic acid may have some solubility limit in each solvent.
Yeah,
Okay, so what you may be able to do is whether you can also combine the extraction with the crystallization process.
yeah, yeah, true.
If you can make enough tannic acid, and then the sonic acid also can be used for inducing the crystallization, then the crystals are settling in, and then you can keep on extracting more and more in the same solvent.
Okay.
Yeah. So the limit is based on the solubility in that particular solvent in terms of the extraction efficiency, not due to the sonication process.
Sir, by your personal experience, can you please share that totally removing tannic acid from tea, will the tea will be with taste or tasteless?
I'm not sure. That I'm not sure.
Okay. Professor Shirish was also there online. I don't know whether he is there or not. Professor Shirish, can you hear us?
What you can do is I could see that a large number of questions there on the chat function.
Yeah, but all are not possible or not possible to
What you can do is
One
you
of
may be able to put them into a Word document later on,
yeah.
and then send it to me. I may be able to answer some of them, and then we can post them later on.
Okay, okay, okay.
Okay, so I'm sure we cannot answer all of them, but at least I can try to give some responses.
One of scientist Professor Dr. Vadgaonkar sir is there, who is having very close association with me. Vadgaonkar sir, do you have any question? Vadgaonkar sir, do you have any question? Okay. Sir, last question we will take. Last question because
ये थोड़ा किसी को इस वीक में एटलीस्ट करा दीजिए फॉर्मेट करा के उनको
okay, okay. So I must thank Professor Ashok Kumar Muthupandian sir that he finds time to share his knowledge for this AICT Atal Faculty Development Program and join with us. Sir, definitely, I am sure that every participants will take advantage of this, and your talk was very fruitful and very excellent. Thank you very much, sir, for joining with us. And please share your PPT with me so that I can share it with all participants so that
Yeah.
they can directly have an advantage of the PPT.
So if I can take 1 minute just to kind of give you the answer for, I can see some answers for the ultrasound scanning.
Yeah, yeah, please.
Ultrasound scanning is very safe because they don't generate cavitation in your body because they are using very high frequency. So when you use, for example, 10 megahertz, at 10 megahertz, you will not have time for the bubbles to form, so cavitation will not happen. But in addition to that, they are also doing pulsed ultrasound. So they will send 10 microsecond pulses with long break so that the bubbles will not grow. For imaging your body, you don't need cavitation. It's like, sound waves are going in, get scattered. And then you look at the scattered sound waves and then build the image. So it's quite safe to do the ultrasound imaging because you don't have you will not generate cavitation in your body. So that's the answer that I would like to give. But also, I would like to thank Professor Sonawane for organizing this session. I enjoyed, presenting this. I'm hoping that.
Sir, last question. Sir, last question. I will interfere.
Yeah.
Sorry, sorry.
Sure.
Sir, it's an interest of my in research field. Sir, can we synthesize self-assembled same assembled nanoparticles using ultrasound, because why I'm asking this question, because ultrasound creates local heating while synthesis of nanoparticles. So can we synthesize self-assembled and nanoparticles using ultrasound?
Yeah, currently it is being tried by some group here in Melbourne. It is possible.
Okay.
I can try to send you some details later on by email.
Yeah. Sir, thank you very much. You want to
Thank
say
you.
something?
No, I just want to thank you, and hopefully, when the COVID restrictions are over, hopefully I can visit you and do the GIAN lectures.
Yeah. So we are excited to see you at VNIT Nagpur in India for our JN program, sir. And
Thank you.
that time,
Thank you, everyone.
yeah.
Thank you, everyone.
Okay, thank you, sir. Thank you.